Namaskar dear students, welcome to you our channel Bio by Gulab Sir. As a part of series of lectures, today's topic is deoxyribonucleic acid DNA. It is very interesting to know about a vital biomolecules without which life is not possible. It has been evolved since immortal time. It took millions of years to get shape up within and outside. Within itself got evolved with the message to produce a machinery of living being, to control that machinery, to reproduce that machinery. Everything is all around DNA. DNA is a self-replicating material. As we say that God is स्वयंभू, वो स्वनिर्माण होता है, वैसे DNA is स्वयंभू. It replicates on its own, it produces on its own. So can be said as a divine biomolecules or a divine molecule. It is really interesting to know about. DNA is a double helix structure. It is being discovered and described by Watson and Crick. They have been described this structure on the basis of X-ray diffraction data given by Wilkin and Franklin. That is, I have mentioned in notes, you can go through. So Wilkin and Franklin, they produce X-ray diffraction data on the basis of which that Watson and Crick got success to describe the DNA structure, and that structure is being widely accepted throughout the world. It is so accurate also. These double helix structure, that what I have shown with a simple line to have best understanding, that suppose this is one helix and this is another helix, has 5-3- and 3-5- orientation. What is 5-3- and 3- orientation? I am going to explain. But before that, how to have that complete knowledge about DNA as mentioned in this diagram. Before going through this diagram, have some basic knowledge about DNA double helix structure. Actually, DNA is a polymer. It is a polynucleotide. It is made up of many nucleotides. So what are that nucleotides then? That nucleotides are made up of nitrogenous base, pentose sugar and phosphate. These nitrogenous base, pentose sugar and phosphate, nitrogenous base join with pentose sugar by N-glycosidic bond. Which bond? N-glycosidic bond. And phosphate join with pentose sugar by ester bond. So this complete structure, that nitrogenous base, pentose sugar and phosphate is nucleotide. So these nucleotides join each other in a chain. So here you can see, suppose thiamine, pentose sugar, phosphate is one nucleotide. Cytosine, pentose sugar, phosphate is another nucleotide. Guanosine, pentose sugar and phosphate is another nucleoside. Now see, these nucleotides are joining each other by ester bond. Ester bond is present. For example, if I have to give here, this is what a ester bond. That what a ester bond present between. So each one has ester bond in between. So these nucleotides join with another nucleotide by ester bond. And hence many nucleotides in a chain form a helix. This is a double helix. So DNA is a double helix structure, two polynucleotides. So this is one polynucleotide, this is another polynucleotide, they arrange anti-parallel to each other. Now that twist is 36 degrees, that is what mentioned in notes. Now we have to know how to explain this DNA structure. See, this is a double helix structure, you can draw this way, having nitrogenous base, pentose sugar and phosphate in a chain. To know how to explain, so step by step we can go. DNA is a double helix structure. When word come helix, then what is helix? Helix are made up of nucleotides. Then what are nucleotides? Each nucleotide is made up of nitrogenous base, pentose sugar and phosphate. Then word come nitrogenous base. Then what are nitrogenous base? Nitrogenous bases are of two types, purines and pyrimidines. Purines and pyrimidines. Purines are adenine and guanine, and pyrimidines are cytosine, thymine and uracil. Then how they are combined, how they are formed pairing. Purines, adenine and guanine. Adenine always pair with thymine, as shown here. Adenine always pair with thymine and guanine always pair with cytosine and vice versa. That cytosine combined with guanine and adenine combined with thymine through hydrogen bond. Now these nucleotides on the same helix combine each other by phosphodiester bond, and two nucleotide and two double helix, they are separately combined by hydrogen bond, and thus two helix come together because of this hydrogen bond between that nucleotides on two different helix. So once again, DNA is a double helix structure. Each helix is made up of nucleotide. Each nucleotide is made up of nitrogenous bases, pentose sugar and phosphate. Nitrogenous bases are of two types, purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are thymine and cytosine. Adenine always pair with thymine with double hydrogen bond. Guanine always pair with cytosine with triple hydrogen bond, and vice versa. And because of they always pair with adenine, always pair with thymine, vice versa, guanine always pair with cytosine, vice versa, these pairs are called complementary base pairs. Please remember, what are complementary base pair? That adenine always pair with thymine and thymine always pair with adenine. That is what. Guanine always pair with cytosine and cytosine always pair with guanine. Therefore, they are called as complementary base pair. So, with the formation of hydrogen bond between complementary base pair, two helix come together, but they are anti-parallel. They run anti-parallel. They are parallel, but anti because they have twist of 36 degree, and that is what I have mentioned in notes, that you can go through these notes, you will find every sentences are there. See, as a point of reference, what I told you, that Mason and Crick, they have described on the basis of data, that is what X-ray diffraction data given by Wilkin and Franklin. DNA is a double helix structure. Each helix is made up of nucleotide. Each nucleotide is made up of nitrogenous base, pentose sugar and phosphate. So nitrogenous base are of two types, purines and pyrimidines. Purines are adenine, guanine, and pyrimidines are thymine, cytosine and uracil. Uracil is not a part of DNA, it is only present in RNA. Now adenine always pair with thymine. Guanine always pair with cytosine, therefore they are called as complementary base pair. Adenine always pair with thymine by double hydrogen bond, and guanine always pair with cytosine with triple hydrogen bond. These nucleotides on each helix are joined to each other by phosphodiester bond, and nucleotides between two parallel helix are joined by hydrogen bond. Each loop, that is what is loop? See, this is what one loop from here to here. That is half circle and half circle, complete circle, that is one loop. So each loop is made up of 10 pair of nucleotides. These are the 5 and 5 plus. That 10 pair, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, completing one loop. That each loop consists of 10 pair of nucleotides, means total 20 nucleotides. Now the subsequent, that is distance between two nucleotides, is 3.4 Armstrong. Therefore, length of one loop is 34 Armstrong. That can be said in nanometer, 3.4 nanometer. And the width of DNA is 20 nanometer. That is clearly mentioned here. And the twist is in 36 diagram, 36 degree in that diagram. So you can, with the help of this simple diagram, you can guess this complicated structure. Now, how to have arrangement of this nitrogenous base, pentose sugar and phosphate is clearly mentioned in this diagram. That need to be practiced. Very important is that. What are 5' and 3'N? To understand this, I have this another formula based explanation. See, pentose sugar which are involved in the construction of this DNA, that they are oriented in opposite direction on two double helix. Here the orientation is this direction and here orientation is this direction. Now see, with this pentose sugar first carbon that bond form with nitrogenous base, therefore we have to count from here 1, 2, 3, 4 and 5th carbon from this side. From this side 1, 2 and 3rd carbon from this side. Now why this 3rd carbon is not free? Because it forms a ester bond with S2PO4, and here 5th carbon form ester bond with phospho or S2PO4. So with the loss of water, so if to understand here, with the loss of water, that means to understand, let you know, here this water molecule get lost. Now when it lost, I rub it as it lost, H2O get minus, So naturally the ester bond formed between these two can be shown like this. So here this is the ester bond. Here again, how ester bond formed? Again loss of water. See, so since that water is lost, as I told you, all the polymers, they are produced with the loss of water. Now see, this is phosphodiester bond formed between two subsequent nucleotides. This direction also same, that phosphodiester bond formed between two subsequent nucleotides is like this. Here, here also that phosphodiester bond will form between these two like this. So you can guess phosphodiester bond between these two subsequent nucleotides and two helix. This shows that which carbon is engaged. Now, if that chain is end from that direction, first, second, third, fourth, and fifth carbon will be free. From this direction, third carbon is free. Therefore, this helix is in orientation of 3', 5'. If this is the first pentose sugar and this is the last pentose sugar, here you can see fifth carbon is free and here this third carbon is free. Therefore, this helix has 5', 3' orientation. So, I hope you got it. What does 5' and 3' means? Fifth carbon of that pentose sugar is free from the direction is called 5' and third carbon of that pentose sugar is free from another direction is called 3', and hence the orientation is 5', 3', 3', 5' as shown here. 5' has that orientation, here this 3', 5' and here 3'. It can be written in that way as it is supposed to be. So here 5' will have opposite 3' and 3' will have opposite here 5'. And here it is clearly shown. I hope you did understand DNA double helix structure. These are the notes given, for that you can note down, and even you can have reference from internet with this structure which I did explain you. These are the structure that will help you to get clear idea. Even if you are in doubt, you can ask it in comment section. Also, I would like to appeal you to share and subscribe our channel. Thank you. Enjoy.